Dear student, in this video, we study the last point of this plant tissue culture, unit number 3, protoplast culture, cybrid and somoclonal variation. The protoplast, it is a naked cell surrounded by a plasma membrane. It can regenerate cell wall, grow and divide. Is a fragile but can be cultured and grow into a whole plant. Cells can originate from any type of tissue. It is important for crop improvement. Via somatic hybridization, new hybrids are produced. It is used by biotechnologists, microbiologists as well as pathologists. Somatic hybridization was first introduced by scientist Carlson in Nicotiana Gaulia. In 1960, E.C. cooking scientist contributing to the enzymatic isolation and culture of a protoplast. Protoplast refers to the separation of protoplast from plant tissue. Important to isolate viable and uninjured protoplast as gently and as quickly as possible. And for this isolation of a protoplast, which is viable one, uninjured one, and which is gently as well as quickly, there are two methods. In the one method is mechanical method and second method is enzymatic method. We first study mechanical method. In this method, tissue is immersed in 1 molar sucrose until protoplast shrunk away from their enclosing cell wall. That process is termed as a plasmolysis. Plasmolysed tissue is cut with a sharp knife at such a thickness that only cell walls are cut. Plasmolysed cell. Undamaged protoplasts in strips are released by osmotic swelling when placed in a low concentration of sucrose solution. Problem encountered during this technique is some cells released uncut, which is having complete protoplast, while the rest produce broken dead protoplast when there is a low sucrose solution is present. In this enzymatic method, number of enzymes are used to dissolve the cell wall for releasing protoplast. After that, the incubation of leaf segments overnight in enzyme solution, then the mixture is a filtrate and it is a centrifuge. Then the protoplast, it is a forms in the form of a pellet. Then that pellet protoplast is washed with sorbitol and recentrifuged. Then we can obtain the clean protoplast, which is a float. These protoplasts, these are pipette out. Then the two enzyme mixture, that is mixture A and mixture B, these are used one after the other. Then this leaf segment with mixture A, which is containing macrozyme in manifold at pH 5.8, are vacuumed, infiltrate for 5 minutes, transferred to a water bath which is having temperature about 25 degrees centigrade and subjected to slow shaking. The enzyme mixture is then replaced by fresh enzyme mixture A and leaf segments are incubated for another hour. The advantage of this enzymatic method is in this manner. It is used for variety of tissues and organ such as fruits, roots, petiole, leaves. Osmotic shrinking is minimum. Cell remain intact and not injured. Protoplast radially obtained. And in these enzymatic methods, there are two methods involved. One is nothing but a direct method, which is a one step only, and second is nothing but a sequential method, which is containing two direct method. The mixture is filtrated using nylon mesh and centrifuge for 1 minute. Then it is washed 3 times with 30% mannitol. The cells are then incubated with enzyme mixture B, which containing the cellulose in a mannitol solution, which is having the pH 5.4 for about 90 minutes, and the temperature is 30 degrees centigrade. The mixture is centrifuged for 1 minute so that protoplast form a pellet and clean 3 times with sorbitol, and it is clean 3 times with the help of sorbitol. Then the purification of that protoplast is essential, and that purification of protoplast is done by removing undigested material which is present in it in the form of a debris as well as burst protoplast. Then these debris are removed by filtering the preparation through a nylon mesh. The enzymes are removed by centrifugation, whereby the protoplast settle to the bottom of the tube and the supernatant removed with the help of a pipette. Intact protoplasts are separated from broken protoplasts through centrifugation and removed by pipette as they are collected at the top of a tube. In this protoplast culture, isolated protoplast can be cultured in an appropriate medium to reform cell wall and generate callus. The optimum for this, the optimum culture conditions, these are necessary, and in them the optimum density of that temperature is also essential. Optimum auxin to cytokinin ratio is essential. Glucose and sucrose, these are present in that culture medium. They can be maintained osmoprotectant in that medium. Then temperature is ranges in between 20 to 28 degrees centigrade, as well as pH is also maintained in between 5.5 to 5.9. The casein hydrolysate, it is a present, and it is a present in a 25% along with BAP and NAA. Protoplast culture in a suitable nutrient media first generate a new cell wall. The formation of a complete cell wall with a wall is followed by an increase in size, number of cell organelles, and then it is incubated for cell division. The first cell division may occur within 2 to 7 days of a culture, resulting in a small clumps of a cell, also it is known as a microcolony. Within 1 to 3 weeks, these microcolonies, these are occur. From such a clumps, there are 2 roots to generate a complete plant, and that complete plant, they are depending again on the which type of that species. Plants are regenerated through organogenesis from callus masses. The microcalli can be made to develop into somatic embryos, which are then converted into whole plant through germination. In this protoplast culture, it is having certain importance, and that important is again along with that protoplast culture, we can be easily study of osmotic behavior, study of that IA action. We can easily study the plasmolemma, then how the cell wall formation is occur, it is also studying. Then organelle isolation is done. Morphogenesis we will study. Then virus uptake and replication, how it is takes place from that protoplast, it is also study. We can easily study the photosynthesis which is takes place, as well as a gene transfer. Means from these protoplast culture we can study these various points, therefore this protoplast culture getting certain importance. Then there are various factors which affecting the protoplast culture. In that, first factor is nothing but that plant species belongs to which species and varieties. If in them small genetic differences is present, then it is leading to varying protoplast responses to that culture condition. Then the plant age and which organ we selected, it is also important, playing important role. Age of the donor plant and its development. is also very important one, and that is nothing but a stage are germinating embryos, plantlet, leaves. These are directly affect on that plant age and organ. Then third point is coming pre-culture condition. When we doing this protoplast culture, it is highly influenced by the climatic factors, and a different culture of seedlings yields protoplast which is having which is giving different responses when these are cultured. Pre-treatment of that tissue before isolating that protoplast, it is also playing important role. Cold treatment, plasmolysis and hormones, they increases the chance of recovery of viable protoplast and their plating efficiency. Then density, it is again an important factor. Then crucial as it influence plating efficiency and surviving of a protoplast. At higher density, protoplast compete with one another, the protoplast compete with, or in them competition we observe, while at lower density losses of metabolites from protoplast is more. Means these are nothing but various factors which is affecting on that protoplast culture. Protoplast fusion technique. There are, means when we isolated that protoplast which is belonging to the same species or different species, and we have to fuse these protoplast for the production of a next generation. Then for this protoplast fusion there are various techniques. First is nothing but electrofusion. Second one is nothing but polyethylene glycol induced fusion, it is simply termed as a PEG. And third is nothing but high calcium ions and high pH technique. First we study the electrofusion. Here mild electric stimulations is being used to fuse protoplast. Two glass capillaries, microelectrode are placed in contact with that protoplast. An electric field of low strength give rise to dielectrophoretic pole generation within the protoplast suspension. This leads to pearl chain arrangement of a protoplast. Number of a protoplast within a pearl chain depend upon the population density of the protoplast and the distance between the electrodes. Subsequent application of high intensity electric impulse for some microseconds results in the electric breakdown of a membrane and subsequent fusion. There are two types of current that are used here. First, C current, which is having given the frequencies DC current again, which is means these are different frequencies current, these are used and their application is done, and therefore it results into formation of pearl chain, then it results into formation of a pores, then it results into fusion of that protoplasmic material, and then the two different species their protoplasm is fused with each other, but for that purposes specific current is used, and that specific current these are having the different strength, and it is given in this diagrammatic representation. Second method is nothing but a polyethylene glycol fusion. In this PEG method, this is a chemical has been identified as a possible or potent fusion of that protoplast. It has a high molecular weight, about 1500 to 6000, and usually these solution of about 28 to 50% is used for the protoplast fusion. This polymer binds to the lipid membrane of cells and thus induces the fusion. This fusion takes place for 45 minutes in incubation, and that incubation is rotation time period. Then fusion takes place slowly during elution of PEG with a liquid culture medium. Then which are the applications which is essential for these protoplasts can be used? In that first is nothing but this protoplast isolation that is helpful in the production of a hybrid for somatic hybridization to overcome sexually incompatible species. Then it is mainly responsible for the injecting foreign material into the cytoplasm for DNA transformation, used to study wall synthesis and decomposition, as well as for the studied as a single cell system. For these all purposes, these protoplast isolation methods as well as it is helpful. In that first we going to the point hybrid. What is mean by hybrid? How it is produced? The hybrid contain nuclear and cytoplasmic genome of one parent and only cytoplasmic genome of the second. Means in that hybrid, the fusion of that protoplasm is takes place, but when that hybrid is formed, it contains the nuclear and cytoplasmic genome of one parent. From another parent, it take the cytoplasmic genome of the second. The somatic hybridization fusion of protoplast that facilitates the mixing of two whole genomes and could be exploited in the process at intergeneric, interkingdom and interspecific level. Somatic hybridization is used to produce hybrids from sexually incompatible species. This method could also be used to study selection procedure. Then next, somatic hybridization. There are certain limitations of that somatic hybridization. In that first is nothing but an intergeneric crosses between widely related plants which are not compatible sexually are not possible. In certain wide crosses, elimination of chromosome from hybrid cell is another limitation of somatic hybridization. In a third limitation, the protoplast fusion experiment, the percentage of fusion between two different parental protoplasts is very low. For hybrid identification, selection and isolation at the culture level, there is no standardized method which is applicable for all material. Injection of foreign material protoplast being wall-less show high pinocytic activity and can inject biologically active foreign bodies such as DNA, plasmid, bacteria, viruses, etc., and it results into modified cells. Advantages to plant breeders in getting more efficient crop varieties in near futures. There are various advantages of protoplast fusion. In that first, it facilitates the mixing of two genomes and can be used in a process at interspecific, intergeneric or even intraspecific levels to create new strains with desired properties and for strain improvement. Mixing two genomes open the door to gene transfer and a study of gene expression, stability of several traits and cell genetic changes. There are some disadvantages of that protoplast fusion. In them first, during the mechanical method of isolation of a protoplast, it yields a very small amount of protoplast after a rather tedious procedure. It is not suitable for isolating protoplast from meristematic and less vacuolated cells. During and subsequent to digestion of the cell wall, the protoplast becomes very sensitive to osmotic stress. Thus, cell wall and protoplast storage must be done in an isotonic solution to prevent rupture of the plasma membrane. In this protoplast fusion and culture, we have to study what is the future. Up to now, transfer of useful genes from one species to another. Technique used in strain improvement for bringing genetic recombination. This technique used in microbial engineering. New strain produced for industrial purposes. Here, protoplast fusion used by molecular biologists, biotechnologists, tissue culturalists, industries as well as alcohol production. Creation of new strain of Saccharomyces from Saccharomyces cerevisiae and T. raceae. Alcohol production up to 80% from cellulose material, less dependent on fossil fuel. The strain from Saccharomyces euryarem can produce low carbohydrate beer of acceptable flavor and production of bitter beer. Here, in crop production also it is playing important role. In that, first is nothing but a palm oil production. Largest source of edible oil become before soybean oil in the world, contributes 31.8% of the world production of oils and fats. It takes 8 years to fully grown and produce fruits. Protoplast culture helps to grow the trees faster to satisfy the increasing demands. Plantlets obtained within 2 years. The example is citrus. In that citrus, subtropical fruit crop in the world, and here citrus sinensis and citrus reticulata. From them, the new seedless variety of citrus fruit, that is nothing but a hybrid, is produced, which is faster than the breeding program. Date fruit production as well as Diglett Nor and Tuckerbout, the two varieties of date fruits, these are produced by using the combination of a different two varieties, and it results into formation of a new strain which is occur only due to protoplast fusion in case of a date fruits. Here it is also applicable in potato production. Solanum tuberosum and Solanum brevigenes, these both are incompatible to each other, but their protoplast fusion, it results into production of that potato variety which is showing resistant to potato leaf roll virus. This hybrid shows resistance and were female fertile one. In conclusion, we found that in a nutshell, protoplast culture has found its many uses and application in field such as genetic engineering and crop breeding, genetic transformation by introduction of a transgene DNA, somatic hybridization by protoplast fusion of species, or subspecies resistance to traditional crossbreeding and isolation of subcellular organelles are all example of how the development of protoplast system has led to an increase in the versatility of plants. Thank you.